everybody, welcome to another episode of the Labiotech Hangouts. So this month we have the co-founder of Microbiome here. Hi, Thomas.
Hi.
Hey.
Hi, Philip. How are you doing?
Good. Yeah, in our new office, it's not completely mounted yet, but yeah, it's cool. And apparently it looks like a studio
over there.
Cool.
Great.
And good it worked out. I think you were busy in the last few months. You're in a digging mode, as you told me. So good you're back and can talk. And so just a quick sentence about yourself and the company. So Microbiome, you're a microbiome startup based in Zurich. We specialized in isolating, characterizing novel intestinal microbes, and created in 2015. And you raised, you told me, 2.5 million until now for dilutive and non-dilutive. And I think to start off, I think you have a really interesting story as a founder. As you are an engineer, you worked in microbiome, and then you basically started the company, but without your own technology initially, and it's not the technology from your own work. So can you go back into that on what were the reasons for it, and how did you arrive to where you are today?
Yes, so I'm an engineer by training, food biotechnologist. So I have been working on bacterial biotechnology since my master's, and I had the pleasure, the honor to go to Jouy in 2007 to do my PhD at the epicenter of microbiome at the time when
just next to Paris.
Yeah, in Joël Doré's lab. Yeah, you know Jouy very well since you grew up there. And to know this field closer at the moment where big discoveries were being made. Still, I kept the engineer in the heart, and I saw need also for technology. And after my PhD, I came back to Zurich, to my alma mater, the ETH, which is an engineering school, mainly to learn to isolate this bacteria and to understand their interaction in fermentation models. I found myself between the clinicians that are actually across the road, here over the road, IBD, gastroenterologists we were working with, the biotech people here at ETH, Christophe Lacroix, that co-founded the company. And we decided that something needed to be done, namely isolating this bacteria and starting assembling mixes of them based on their functionality. And this functionality, being driven by functionality, is something that I learned at Jouy in Joël's lab, that the key to the ecosystem is function.
Okay, makes sense. And so that's great. And so how did it go from like 2015 when you started, and where are you standing right now?
Yeah, we started with the idea, which is in biotech, it's quite courageous to start just with the idea. We got from ETH the possibility to be an incubator, so they have a specific incubation program. Yeah. We got a year to work out our idea here at ETH in the ETH labs, and we managed to have a proof of concept from scratch in 1 year, where we isolated and we found a way to do mixed cultures of bacteria and prove our point and prove our hypothesis. And since then, we spun out of ETH and have, of course, close research collaborations with University Hospital here, with ETH Zurich, but are independent and have been working on the tough part, which is bringing the proof of concept towards clinic. So the whole preclinical considerations, the production considerations. Don't need to tell the biotech community that the idea is the easy bit before you go to clinics.
The execution is the hard part. Yeah. That's great. About the ETH is great. We'll get back to that later on the kind of incubation. I think it's very rare and they need more of that in Europe. But maybe now on the technology. So you said about it's really hard to identify new microbiotes or characterize them. Like what's so difficult about it, and what makes your technology?
So for our main idea was that the common denominator is function. And the research we have done since then has shown us that the smallest biologically relevant unit in the microbiome is not the bacteria, but a mix of bacteria that can encompass a function. And based on that, we started developing initially to mimic FMT, but later on then also to reverse different forms of disbalances. We started to find ways to co-cultivate this unit, this mix of bacteria, in a way that can be then used for in vitro validation, but also for production. Because one of the main challenges of this new modality, living bacteria, is to first bring it viably into humans. So you need to exert your function in humans, but also an often forgotten point is the GMP production. I mean, there's people that are specializing now. There's companies that are dedicating themselves to that. But having a process where you can reproducibly produce larger batches of such bacterial mixes is a challenge we have been working on for 3 years now, and we think we have a very unique solution to that.
Yeah, that's really interesting. So really looking at the function of the microbiome, a bit like a mix from a fecal transplant, but trying to recreate it and control it, which is in-house.
The fecal transplant actually transfers function by redundancy. So you have several.
Well, I guess you have several functions, like not just one.
Yeah, of course. Yeah. So you have we work mainly. You have a functional scaffold that holds the whole thing together, where you have degradation of fibers, some intermediate metabolites that end up in the end metabolites everybody's talking about, and then have a big effect on physiology. Now, if you can you get back to the question? Wait a second.
Yeah, it's really on the how is it different from a fecal transplant, basically?
Yeah, exactly. Now this functional scaffold is in every fecal sample in a high redundancy, so you increase the probability of transferring these functions. Now, if you want to move this towards a defined drug, you need to guarantee functionality with every transplant. And you need to make sure you have the functionalities you want in there in your transplant. And that were our boundaries we started with. What we saw in our work, in our lab work, is that many of these bacteria only grow in a certain context. So since you have an ecosystem just like a rainforest, you need upstream and downstream of each bacteria. You need somebody that provides substrate and somebody that makes sure there is no self-inhibition by the products. And based on that thought, we started doing research and hard work, actually, isolating, combining, and the work paid off. We got a very robust a minimal set of bacteria that is working very robustly in preclinical models, and that we want to push into clinics starting next year. So
that's great. So basically, if I summarize, is really you have some company I'm trying to put you in the microbiome space. You're really companies doing like one single bacteria or functional bacteria, life biotherapeutics. You have the fecal transplant with all the different bacteria, and you are kind of in the middle, kind of controlled, but still diverse and more functional.
You can say that we developed a technology where you can combine the functions you want in we call it supernatural way, because you can choose, you can have different types of microbiomes combined, but also in a simplified way, so you can make sure that you control your product. So in the microbiome space, we try to bring together the full control of the single bacteria approaches with the ecologically rationale of fecal transplant.
Yeah.
But being aware that you need something that you need to be able to produce reproducibly, that you need to be able to guarantee efficacy. And in our research, what we've seen is that efficacy is very strongly linked to the viability of your bacteria. And how you make sure that the functions you want to transfer are actually exerted in vivo.
That makes sense. And so now you're talking about application and moving into clinics. So so what's your plan on that? Like moving one specific function to clinics? And
the final, the proof of concept is a very, very nice proof that our initial hypothesis is worth pursuing. Now the final confirmation is the clinical confirmation, and that's of course what we want to do. We have a compound on ulcerative colitis that we have been validating extensively, and where we have a mode of action in order to feel that we're at a point where we can go into translation and want to push this compound into clinics. We mapped out the production process and are now ready, starting fundraising during next year to get our clinical program financed.
Okay, and then you plan to start the clinics basically next year or something around that?
Maybe. It's always dangerous when you're fundraising to say a year. We tested on the relevant models to feel confident the translation will work, and that we feel. We mapped out the production process and are currently looking at how we are going to produce this GMP, with whom, etc. That is one of the key risks we identified and we have been working on. And at the same time, we're mapping out the clinical implementation plan. Means reaching out to regulatory agencies. To our knowledge, we're the first mixed consortium that comes out of a mixed production. So we want to push this towards the regulatory agencies to find out where we can go first in human.
That's good. Okay. So basically soon. It's good. And
as soon as possible.
That's a good answer.
So depending on your willingness to fund us, we can make it happen as soon as possible. But the main risk we saw is really how do we get a robust production process, and that we have very well mapped out.
Sounds like a common challenge for microbiome and also for gene therapy. Any complicated biological product, it sounds like it's a big challenge. And maybe talking about the fundraising, so you plan to start a fundraising soon. Who are you talking to? Like, what's your target? What's your plan on the fundraising side?
On the fundraising side, we have the next two big milestones are obviously the first GMP batch and then the first dosing in man. We have also other compounds in early stage we want to pursue. So we go for a pipeline approach where we leverage the knowledge that we have generated in follow-up indications. You know as well as I do that clinical validation needs quite some resources. So the obvious suspects would be people that are very knowledgeable in the field and have the resources to carry on through the different clinical phases and to support further developments of the platform.
So typically a VC like Seventure or someone like this.
Yes, Seventure is obviously the biggest VC in the space. Depends a bit on their portfolio approach. We obviously we are talking to different VCs in the field.
Yeah. And I guess they know Ina and Joel really well as well. Okay, and just from the side, I guess you raised 2.5, so you want to raise several millions in your Series A.
Yeah.
What's the size?
The size is a low double-digit million. Yeah. Depending, I mean, tranching is then a question of further discussions. But now we need the resources to. We're currently in an incubator, so we have limited space to grow. So we need the resources to grow, to go into clinics. That will be the biggest chunk of the financing will go into the lead compound validation and bringing it into clinics. But we think that our expertise on the technology and the assembly of consortia for different indications is something worth pursuing. We have some collaborations on that side, and that will be a part of Pharmabiome in the future as well, to stay on the edge of understanding of the microbiome, and to which we did already with follow-up indications, to go from the analysis of biomarkers to the assembly of a consortium to the biological validation of mode of action.
Yeah, that's good. Well, and I guess for the space, once you raise the double digits Series A in the millions, you can find a bigger office space.
Yeah, actually, we're quite comfortable. We're 8 people in an office, so it facilitates the very interdisciplinary communication we have to have at this early phase. So we have data science, biotech, microbiology, business development, and the startup is an iterative process. So keeping everybody on the page is easier at the moment in small offices.
I totally understand. We are 10 people now, so I totally understand.
How much space did you get in your new office? Are you planning to double the number yet?
Enough space, but no, enough space. But it's really hard to find in Berlin at the moment. The space. It's anyway. It's another topic. Yeah,
it is
good. So, and maybe one question on the strategy. I think because when we talked last time, I think you were also, I mean, more on a top level, you really have a platform technology which could be applied for your own products, but you can also partner with other people. So what's your overall strategy or more vision on the 3 to 5 year plan? Do you plan to have like a really broad pipeline or single asset, or what's your vision really?
No, we of course. So the versatility of our approach, of course, calls for a broad pipeline of different assets, which we are working on and which we also have intellectual property filed on. Now, it is also our understanding that your first indication is the value driver and the proof of the validity of our approach. We have seen lots of partnering in the space in the last years, which is something that we are considering. We're also discussing with pharmaceutical companies. The question is always, what moment do you want to start partnering, and is the possibility there? If the possibility is not given, we are also willing to bring the compound into clinics ourselves. A phase 2 partnering would then obviously be something that you would actively work towards.
Okay. Yeah. So wait as long as until you have efficacy.
We don't see us. No, but we don't see us. We think the microbiome field is in a very early stage. The importance is to be science-driven and mode of action-driven that we worked on. Partnering is a very good way to bring our compounds to patients, but we don't want to be a classical platform in the sense that we just develop and then partner immediately. We want to bring our products to the patient.
Yeah. Makes sense. I mean, and you capture more value at the end, so it makes also sense. Oh. It's good you mentioned the microbiome field. I wanted to talk with you on getting your vision of the field a bit broader perspective. It's definitely a super hot field, but it's been hot for the quite past a few years now.
Yeah.
So maybe I don't know, what do you still see missing beside a product on the market?
From our point of view, the big discussion, biomarker versus causal mode of action, is still something that we as a company spend much time on. So we have in our mission to understand mechanistically how the microbiome affects the host. An early field always gives much space for projection and speculation. I think in the next few years we will see which therapeutic approaches make sense, and we're very happy to contribute to that. We think there is different propositions from our competitors that all make sense in their way. Yeah, and that might need to be combined. What our conviction was from the beginning is that no matter what the solution will be, people will need the technology to deliver, and that's why we have been working on that since the beginning.
That's good. I mean, at the end, the data in the clinics will talk. So,
yeah. And it's a huge field. So several therapeutic approaches will be in the market in the end.
That's good. Cool. And maybe we can last question more on Switzerland and ETH. I mean, why besides going there for like you went there for expertise on bacterial cultivation, but what was your reason for going to ETH? Like, did you specifically choose ETH to start the company, or
I came no, I didn't come to start a company, even though Metagenopolis was founded during the end of my PhD. So I saw this translation happening in Paris already, and Mad Pharma was founded at the time. The result of my PhD research was that to handle the microbiome, you have to think in multiple layers, bacterial antigens, but also metabolites, interaction with the metabolites, etc. So I came here because I was interested in the model Christophe Lacroix had, where you can actually maintain a complex microbiome in a bioreactor and take the host compound out and start understanding the ecological mechanisms. It was purely scientifically driven. We went to tech transfer to say, we have this idea, we want to found a company, do this next to our postdoc, blah, blah, blah. And they told us, they brought us down to earth, and I'm quite grateful for that. I said, you can't just found the company on a 20% basis. Either you go for it or you leave it. And that's when I found myself on the right spot at the right time between clinics, biotech, and basic microbiology. And from my experience in the microbiome field, I saw this as an opportunity to make a difference and to develop something that makes sense for the patient and for the scientist, and increase the potential of the microbiome. A frustration that stayed with the probiotics. I mean, the probiotic field was a bit scientifically frustrating because the idea is valid, but then
there's no data. No,
there's no data.
No, it's good you make a good comment on the tech transfer offices. It's pretty rare. I don't hear that very often. Maybe can you?
I didn't comment on everything we did with them, but that point was a very good eye opener.
But maybe you can have a more broader comment also on the kind of entrepreneurship ecosystem or biotech entrepreneurship ecosystem in Zurich or the rest of Switzerland. How do you feel about it? Like,
the biotech ecosystem in Switzerland is obviously big, since there is big pharma not far away, and there is a few very good examples of translation. I think ETH is very special because they have this engineering mindset where translation and application is obviously very important. And
MIT.
Yeah, and geographically, we're currently hosted in ETH Center, which is right next to the university hospital. So it allows you to be very close to the application. There's much effort in supporting entrepreneurship. There is courses that are from other institutions like VentureKick and similar, that get you from the idea towards a real business plan. So the ecosystem is very, very supportive. Execution is still up to you, obviously. I think in the biotech space, Switzerland is interesting as well because you have very well educated technical personnel as well, coming out of pharma, and this allows you to implement at a high level of quality.
Yeah. No, that's good. That's good. Cool. Then I think it was my last question, just to get a bit of perspective. Maybe a final question is, who should we interview next year? Another awesome biotech founder or young biotech founder in Switzerland?
In Switzerland?
Or in Europe, actually.
My favorite biotech in Switzerland, it's the guys from which one is my favorite? No, I have to be careful with my network.
You can send it to me by email as well. It's a tricky one.
No, it's a very tricky one. But I think in the ETH space there's many nice biotech startups. I don't want to be too academic. I think there is a lot of entrepreneurship besides this purely academia incubated startups that are doing well. Obviously, the biotech space, Tolremo has raised a very nice round last year. And I think Stephanie would be more than happy to have an interview. If she didn't do it yet.
That's it.
On the medtech space, there are quite some nice cases as well.
Cool. I wrote that down. Great, Thomas. It was a pleasure. Thank you very much. And good luck for the fundraising. Good luck for the next clinical milestones. And let's catch up soon.
Hopefully. Okay. Have a nice day.
Thanks. Bye-bye.